Our speaker today is Melissa Deinys. Okay, she is a distinguished researcher specializing in sustainable nanotechnology for environmental applications with a focus on combating plant pathogens that threaten coastal shorelines. She currently serves as a researcher at the Material Innovation for Sustainable Agriculture Lab at University of Central Florida under the mentorship of Dr. Santra. Melissa has a strong commitment to collaboration and works with multiple labs at UCF and numerous external partners, including MRC, demonstrating her dedication to advancing impactful research in the field of sustainable nanotechnology. All right, and with that, Melissa.
So, as you mentioned, my name is Melissa. I am a researcher at the University of Central Florida. Just a little bit background from me. My parents are originally from Cuba. I lived in Miami for my whole life until I moved to Orlando to continue my education. And that is when I joined the Material Innovation Lab, which I currently reside in right now. As I lived in Miami and I'm from the Caribbean, my whole life I've been around shorelines. I practically grew up on the beach or boating or out in the water. So this is a very personal project and it's very close to my heart. So, also a little bit more background. I began my research journey as a specializing in plant pathology. So not necessarily coastlines, but more looking at botany and how these systems work and interact in the field. So first of all, why are red mangroves important? If you're here, I'm pretty sure that you're all very familiar with them, but they shelter native organisms. They're very big for our oyster fisheries, also protecting our coastal communities. Anyone that lives on a beach, or especially here in Florida, know about flooding and how mangroves protect against flooding and those type of impacts that are caused during hurricane or sea level rise. But they also have a lot of economic benefit. As I mentioned, they're one of the key factors for a lot of our fishery industries. Also, there's been kind of studies that show not only what some people come to think as the importance of mangroves, their root structures that kind of anchor soil. The foliage or the leaves of the mangrove also have a very big significance. They stopped winds. So when we have hurricanes and you get all those big winds coming in, the foliage or the top part of the tree also stops that from being able to knock down power lines, cause damage to homes in the area. So kind of the whole system of the mangrove is important. So since the 1960s, we've kind of been seeing a decline in our mangrove populations, not just in Florida, but all over the world. We've gotten to about 60 to 80%, depending on what review you look at or what parts of literature you look at, of our native mangroves have been going away. This is for a lot of factors. You have housing, more people are coming to Florida, we need more housing, also deforestation. But now we're kind of looking at a third reason why these mangroves might be dying out. And that's plant pathogens. So as I mentioned, my background's in plant pathology, but for those of you that are kind of not familiar with the subject, there's kind of two main classes of plant pathogens. You have the whole family, which is like bacteria, viruses, anything that could kind of cause disease in humans can cause it in a plant. They're just another field. But you also have epiphytic, or what live on the surface of a plant. That is very visual. You can kind of tell something's wrong with this leaf. But you also have it being systemic. So this leaf, I actually tested it, and it does have the pathogen we're going to be talking about later on. But to the naked eye, you really can't tell. Mara came up with a very nice kind of way to say this, is like when you have chicken pox, it's like within you until something stresses you out or something, you'll start seeing the symptoms. So this is kind of what we're going to be talking about today. So symptoms of infected leaves. These are all mangrove leaves I've collected out in the wild in different sections. You kind of have this fluorosis, which is chlorophyll die-off in the leaf. You can see that's yellowing. You have these leaf spots that are uniform right here. And then you also have these necrotic lesions. Now, out of all three of these, the necrotic lesions are what's the hardest to identify as a plant pathogen, because when looking at it, you kind of might think it's either an insect, it could be just like spiders, beetles, a lot of things eat mangroves, sometimes even deer. Deer really like mangroves. I learned that a couple weeks ago. So out of all three of these, you need to identify that it is a pathogen to be sure. But these are kind of visual tells. Another thing that we've kind of come to a hypothesis about is when you turn a mangrove leaf, you're gonna see these black spots on the back. And even when I was growing up, I kind of looked at these black spots as something normal about a mangrove. I'm just like, it's just part of it. I didn't really think about it. When I joined my lab, they were, first of all, chemists. So they think about situations and things differently. One of my advisors, Dr. Jorge Pereira, looked at these and there's like, Melissa, this is not just a normal characteristic. Like, why do some mangroves have it? Why some don't? We need to look further into this. And then when looking at under SEM, which is a special type of microscope, scanning electron microscope, we actually saw that it was stomata damage. So you can see right here, these are normal stomata. They look normal. You can see the two different stomata cells, and these are where the black spots start appearing. So as of right now, we believe it's either how the pathogen gains entry to the plant, or it's how it exits and spreads the disease. But you can definitely tell some of them would be, if you can look right here, I don't know, because it's gray, I don't know if you guys can see it correctly. But if you look at this one here, this one's concaved in, while this one on this side is exploded out. So it could actually be both. But to further confirm this is going to be a lot of testing. But for right now, that's our hypothesis about these spots. So as I mentioned, that little squid looking thing is actually a spore of a fungi. Because right now we're going to be specifically talking about fungal pathogens. And we believe that's how it gains entry into the plant. So now that we looked at the background and what is plant pathology, let's look at what I'm currently looking into now, nanotechnology. So in current years, the last 2 decades, we actually won one of the Nobel Peace Prizes in chemistry a few years back, was actually in nanotech. It's kind of an emerging field. Nanotechnology is very simple, coming from someone that didn't study chemistry at all until joining this lab. It's just, you look at big molecules, you just make them nano. The whole thing is about the size. Because once you get nanoparticles, they're so small, they can actually go into stomatas of plants, so you can reach these diseases that are within the plant. They're also, because they're so precise, you can have them for lower concentrations that would use like a normal fungicide that you would like spray in your garden. You can have lower concentrations and they'll be more effective. So in places like mangrove forests, this is a very key feature that we need to include. Second of all, something I would like to discuss is that all of our treatments are nutrient-based. So let's look at the next slide. So our treatment is consisted of magnesium hydroxide nanoparticles and then normal sodium sulfide solution. For that, we call the magnesium magsol and the sulfur naps. But you're thinking, magnesium is something I take as a daily supplement. How can it possibly be something that can kill a pathogen? Well, when you look at nanotechnology, what the bigger, let's say, imagine this is a magnesium. When you take it to a smaller level, it actually gets new properties. So magnesium hydroxide in its molecular form, it has completely different properties than in the nano form. And the main reason we chose sulfur and magnesium is because they're secondary macronutrients, so the plant actually needs them to survive. But also sulfur is a natural antifungal. And if you guys been out to like the mangrove swamps, you can smell like that sulfur in the air. It smells like eggs. So it's already something that's incorporated into the ecosystem. So we're not reintroducing something that's not already there. So just for the fun of research, the magnesium hydroxide nanoparticles, when I joined the lab, were already made. We had a previous PhD student that's what his PhD work was on creating this magnesium hydroxide nanoparticle. The biggest problem was he used it against bacteria. And bacteria are unicellular. In my opinion, as a fungi person in my lab, they're super easy to kill. Fungi, whole different issues. They're multicellular, they grow faster, they're way more complex. So when I gave them the magnesium hydroxide nanoparticles, how they were originally made, my fungi actually grew more. They ate it, they were like, this is great. So I needed to come up with a solution, and how am I going to turn these nanoparticles into something that can actually be used against fungi? Because I really liked having that magnesium element. That's when we started thinking about hydrothermally treating them, or in very easy words, cooking them in an oven. But it's fancy because it's science. So when you cook them in the oven, you're increasing the crystallinity. The crystallinity is going to be what helps the nanoparticle attack that fungi. It's kind of like, imagine it was a circle, now it's a sphere. It changes the shape of the particle, allowing it for better entry. So we did a bunch of different chemical characterizations. If you're interested in that, we can have another 3-hour discussion about how that's characterized. But for the purpose of this, we confirmed 3 different treatment processes on like different time periods, how cooking them for 4 hours, 12 hours, and 24 hours, just to see what that sweet spot was. We confirmed that the 12 hours, what was best, just chemistry-wise, 12 hours should have killed the most. So we now look at poison food, what actually kills that in the field, because something I've learned, what works in a lab does not usually correlate most of the time. You have to go back to the drawing board and figure out why things are not working. So poison food assay is when we get these 2 different, we make MagSun, which is the combination of the 2. We add it to agar, which is what the fungi eats. We let it solidify, and then we plate the different pathogens onto the plate. The fungi actually has to eat the agar with the solution to grow. So right now you're looking, okay, if the fungi comes in contact and digests our material, will it die? And as you can see, here we have the 3 different thermal treatments. You have 4 hours at the top, 12 hours, and then 24. And you can kind of see the 4 hours kind of worked. The 24 hours also kind of worked. We saw from a lot of growth to kind of minimal growth, but the 12 hours did significantly better. You can kind of see that this sweet spot concentration, you almost have no growth at all. And here we can kind of see like, what would it look on a graph? So this yellow line right here is the normal growth. And then we have our treatments. As I said, there's a very big difference in our treatment to no treatment, but still that 12 hours was the best. Okay, and just because you want to know why it works, especially if now we're going to say, okay, this is meant for mangroves. So why exactly is this nanoparticle killing? Is it just killing because it's super toxic? Because that's also not what we want, because then it'll be super toxic to other things. So this is what we call reactive oxygen species. It's what a lot of studies use to measure kind of, let's say, stress within an organism. How much stress are you causing to that organism, and where is it located? So we're going to look at here, the control. Having the green is the reactive oxygen species that we can look through fluorescence. You're going to have some stress in control. So that's why we do it, to measure what's the difference. Then we kind of look here at the 4 hours. You can kind of see the stress is more spread out. What I would like to mention, just because some of you are thinking it, Melissa, this is super dispersed. And then this is super compacted. Well, is that not going to skew the data? Well, what we found is that all of these samples were prepped and sampled the exact same. So that means that something's occurring to these filaments or the spaghetti structures of the fungi to make it more compact when prepping the sample, which I will get into in the next slide. But as you see, in the 4 hours, we do get more dispersed, but it's kind of dim. And then when we look at the 12 hours, we have the same dispersity of the 4 hours, but with much higher intensity, just causing a lot more stress with the 12 hours. And then, as I mentioned, something's going on with those filaments. Why are they getting compacted? Well, magnesium hydroxide in bacteria have been shown to be able to cause cell membrane damage. So that is what magnesium hydroxide in the nano form does. Like I mentioned, spheres, it breaks apart the cell membrane, things start coming out, which would make sense if we look back at the previous slide. If, let's say, organelles and things are coming out, they're going to want to come together when you spin them, because that's just how it works. Okay, let's test it. Let's see what happens. So we look at how much DNA is leaking after treatment from each of our samples. And as we can see here, the 12 hours had way more significant DNA leakage than any of our other controls. We did the Maxwell by itself, which is what we subtract from. So we do the treatment by itself to make sure that our treatment at this nanometer range, which is what you measure DNA at, also doesn't have a signal, because then you don't want a false reading. But even after doing all those controls and subtracting everything from each other, the 12 hours had great DNA leakage. So, okay, now we know the mode of action. Mode of action is it causes stress, plus it breaks apart the cell membrane, and that's how it kills. But as I said, okay, we tested in lab, we tested what we like to call in vitro, which is like in a petri dish. What happens when you actually apply it to a system, or in this case, a mangrove? So the big two fungal species that cause pathogenicity that we're seeing in mangroves is Neopestilopteopsis and Pestilopteopsis. Those are the two that cause the most or most widespread. There's one other one called Cuvularia, but it's in a smaller subsection. So we want to see the bigger factors first. And these are what we call Koch postulates. You basically get a mangrove, you infect it, wait 8 to 6 weeks, and start looking at the symptoms, which, as I mentioned before, you can see the chlorosis, you can kind of see the starting of the spotting right here, as well as here. So when we did this, what is called this treated infected is we infected these 3 plants at the same time. The treated infected is with the Pestilopteopsis, because that's the stronger one of the two. I was like, if it works on the stronger one, it'll work on the other one. So, but the difference is we waited 2 weeks for this one. So we infected, waited 2 weeks to make sure that the fungi or disease got in there correctly, and then we treated it with our treatment. And as you can see, out of all 3 of them, this one didn't show any signs. I actually have this maker currently, and it still looks great. It still looks nice green leaves. So we were like, okay, maybe we're onto something. Maybe something's working. But also what we looked at is we also found that these diseases can grow on the propagule or the seed of the mangrove. We took samples from the seeds and it was still growing. So that means it can go from mother to seed of plant, which makes sense because mangrove seeds are so big, they take such a long period to develop, they sit on the tree for so long, something systemic eventually will get into that seed. And what we saw is after treatment, we also got full fungal dissociation. Like there was no more growth. Now, one of the biggest questions I get at conferences or whenever I'm saying this is, okay, so now are you affecting the microbiome of the plant? How is that affecting it? Well, our lab mainly focuses on agriculture. They were an agricultural lab before I joined. Still, most of our projects are agriculture. I'm the little environmentalist in the corner, just doing all my stuff. They're very supportive. But the thing is, it's like, okay, you get a fungal infection. What are you going to take? Antifungals. The same time you take antifungals, it's going to do the same thing to your body that this antifungal will do to the plant. That microbiome will regenerate just because what's in that microbiome is going to be in the soil. So recuperate that. But also, I don't know if you see this tiny little growth that's right here. That's penicillium, which is one of the biggest, it's like what penicillin comes from. It's one of the biggest environmental fungi that you'll just find everywhere. So that also shows that we're also not killing all of the fungi that's in the plant. Actually, penicillium is sometimes beneficial to plants. It's just like an environmental fungi. So there was that. but also what happens to the chlorophyll or the health of the plant after treatment. So this was important just because we treat the mangroves for 24 hours. And that's the kind of sweet spot. You get the mangroves, you treat them for 24 hours, and then you take them out of the treatment. So we also wanted to see, are we causing harm that we can't visually see to the plant's chlorophyll that in a year, 2 years is going to have an impact. So after treatment, you can kind of look here. So this is before treatment, the green's before treatment, and then the after treatment is the blue. And kind of across the board, we did the similar. Where we didn't do similar, or where you can kind of see a drop off, is in this nap section right here. You can see that this was before treatment, and then there's a slight drop off after treatment. This is completely normal. You can kind of even see here in the control, there was a slight drop off before and after treatment, and the controls weren't even treated. But what I say is the NAPS is the killing element. So Maxol kills some, but the sodium polysulfide is really what's that antifungal, that strong key ingredient. So you're gonna get a slight bit of damage as you would an antifungal, even if it's so small. But when you look at the Maxol, which is the magnesium section, we actually increased chlorophyll. So when you add them together, which is that last group at the end of the graph, we got it back to the same of chlorophyll before and after. Now, one of the most important tests that we did, and I personally thought was very necessary, is starting to look at model systems of what would be in an environment. So currently we have data for brine shrimp, which is what our crustacean or our crustacean model system, which is actually used by the EPA to test environmental, not environmental, because there's not been an environmental fungicide approved yet, but what they check for fungicide treatments. And usually LD50, which is what you look for, is when you kill 50% of the population. So if I have 10 shrimps, 5 of them need to die, that will be my LD50. LD50 is just a little standard we do. And usually, how we say this, it's like 1/10 of the treatment. So our treatment's at 512 ppm or parts per million. Our LD50 should be 5.12. Usually it's around there. It's usually in the 10 ppm to 5.12 because aquatic systems are so, like, I'm sorry, I'm thinking of the Spanish word. They're so fragile. But so let's look at the components separately, so now that you understand. Our magnesium hydroxide nanoparticle, we actually didn't reach LD50 with the Maxord. We went all the way to 2048 ppm, which is about triple our concentration range. I think more, I think 4 times our concentration range, and we still did not reach LD50. So that means it would need to get so concentrated that it was multiplied by 4 times to be able to kill 50% of shrimp. And at that concentration, I think we killed just right below 60%, if I'm remembering correctly. Now, for sodium polysulfide, that killing element, we did reach LD50, and it was between 256 and 512, so I'm calling it on the safer side around 300. What the 50 would be. That's still above what we treat at. So this was very nice news to see, and I was like, okay, the problem is now we're adding these 2 components together. So we're actually combining these 2 concentrations. We're going to use 512 of the Maxol and 256 of the sodium polysulfide. So let's look here, which is what that is when you look at the toxicity of the 2 components together. This bottom row is the sulfur. This row to the side is the magnesium. And here we can see that we reach LD50 at 256 sodium polysulfide and 1,000 ppm of magnesium. So this means the shrimp have to literally bathe in the solution you're using, which is never what will happen because these solutions won't be, let's say something horrible happens and they're dumped into a lake. The way that it's dumped into the lake, they're going to get so diluted from all that water, they will never reach that concentration. 2, that should never happen because we should be very safe with treatments. Three, let's say we're doing this as a aerial spray, or in a far off future, how we're gonna implement this to wild mangroves. These are gonna be drops of the treatment that are gonna go into the water. So also never reach that concentration. But it also is very important just to see what that concentration is to make sure we never get there. So this has been a very long process, but our next step currently, what I actually have to go check in to the lab in a few hours to look at is we're looking at algae. How does it affect algae populations? Is it going to be algicidal, which sometimes is helpful for algae blooms, or is it just going to be stagnant, like it doesn't cause differences in algae populations? What will it do to duckweed, an aquatic plant? That's also another very important factor. How is this treatment with MRC gonna help mangroves? So, okay, we did all this in the lab, but now the MRC is actually implementing it into the nursery. How does that look? Is it actually helping? What does that look like? And then also how to make, how does Manson interact in the environment? So this is more on the chemistry side, but we're very interested in knowing, okay, nanoparticles dissociate. So what is that breakdown looking like once it's in the environment, or after 24 hours or 2 days? What does that solution actually look like now? Because it won't, I promise you, it won't be magnesium hydroxide, nanoparticles, and sodium polysulfide. It's going to be a whole different makeup. So what is that makeup? And that's kind of what we're looking into right now. But yeah, these are my resources. If anyone's interested, I'll send you almost 100 pages of stuff you can read. And this is my lab. They're very supportive, and they're kind of the reason I've been able to do everything I've been able to do. So, them. And then, yeah, if you guys have any questions, I can hopefully answer what you guys have questions on.
If anyone has no question sheet for me, I'll come grab them from you. Does anyone need a question sheet? Okay. Okay, so we are gonna start with a question from Sue. Okay, so going back to the brine shrimp. So.
I don't think I can go back. Oh. I can go forward, but not back.
Okay, so this test
Yes.
was to see how much solution you could put in to keep the brine shrimp alive, is that right?
Yes.
So see how.
So it's more of
environment.
So it's like more of toxicity.
Okay.
So what concentration would the treatment need to be at to cause toxicity to the environment?
Okay, so you're making sure that the levels that you guys are putting in are safe.
Yeah.
For other things.
So we don't only just want to be able to be like, oh, we killed the pathogen, yay. We also want to be sure that it's safe to be implemented in places or be able to not cause harm to other things, even at the concentration that it kills the pathogen at.
How did you get into doing this research?
Oh, so I went to biotech at Richmond Heights High School, back in high school, once upon a time. And they actually are one of the only schools in the nation that do conservation biology as a magnet program. So it was very unique in the sense that I did research as a freshman in Miami Zoo. Then I went sophomore year, I started at Fairchild Botanic Garden, which is where I continued this research and actually where we found the first signs of this pathogen in Florida. So I've been kind of doing this since sophomore year of high school, and that's how I got into it.
Okay, let's see. Okay. Is the pathogen or multiple pathogens you're looking at, is it something new that you've discovered or has it been around?
So this is just the first time it's discovered in Florida, but actually these 3 fungal families have been shown all over the world to cause harm to mangroves. We have reports from Malaysia, India, China, where you can see in mangrove forest, we have some type of documentation even 50 years ago that they saw these pathogens that were causing harm. So this is just the first time it's documented here, but it's been known through literature that they can cause harm to mangroves.
And does it cause harm to other types of trees or plants?
Yeah, so Pestiloptiopsis is actually one of the biggest plant pathogens that affect agriculture. Also, Cuvularia, the other plant pathogen I spoke about, also huge devastation to corn and other crops. Neophysiloptiopsis in Plant City, where we grow a lot of our strawberries, also is causing a lot of harm to farmers because they're attacking strawberries as well. So plant pathogens aren't just unique to systems or to organisms, they're usually very spread out.
Question about the chlorophyll recovery.
And the ones before that one. Yes.
Okay, so this just looks at how much green
So,
pigment versus yellow pigment.
so it's actually on Amazon, $80. It's a little detector that you can use and it measures it in SPAD units. It takes a little laser, it puts it through the leaf wherever you take it. So it can't really look at the whole general area of it. It's looking at specific chlorophyll, like energy outputs in that area. So usually you take about 3 readings around the leaf to make sure, usually it's pretty uniform. Take 3 readings around the leaf and that's how well the chlorophyll is working in those states. Because if we saw like a big reduction in one of our components, we'd be like, okay, so this is attacking chlorophyll or not allowing chlorophyll to die off. So even if you can't visually tell, we know that there will be a die off or like it'll go yellow in like a couple days or eventually something will happen to the plant.
Okay. Any other questions for me? Okay. Thank you so much, Melissa, and thank you to everyone for coming.
Thank you.